Buenos días, in Spanish, para agradecer la oportunidad de actualizar cuál es el estado de nuestras investigaciones, que desde hace varios años se centran en el papel de un potencial agente desencadenante de la demencia, de la neurodegeneración, que es el virus herpes, y sobre el que llevamos dando vueltas, como muchos de vosotros conocéis, bastante tiempo. Now I'm changing to English. So I will talk about what is the problem with mouse. It isn't working. ¿Sí? ¿Aquí? Sorry. Pero... Vale. Okay. Thank you. Okay. So we are talking to you about why we started to study these things, lysosome system, cholesterol alterations, in the context of Alzheimer's disease triggered or potentially triggered by infection like that of herpes simplex 1. We are a group composed of two branches, a basic branch at the CBMSO and a clinic branch at the Hospital La Paz. We have been working together for many years, so it allows us to start or try to do translational research. Our experimental approach has been maintained during several years, and it continues to seem to be basically the same. We use simple cellular models. The basic one is a human neuroepithelioma, neuroblastoma. Some of the experiments I'm sharing with you have been validated in other models like primary neurons, or in a few cases, iPS-derived neuronal cells. Our hypothesis is that several insults with main interest now in herpes simplex virus, in infection and among infections, herpes simplex virus 1, in the context of oxidative stress associated to the aging process, will trigger neurodegeneration that we follow with studying markers typically associated to Alzheimer's disease, and that we try to dissect functionally by performing transcriptome analysis to try to look at the mechanism linking the triggering with the result. These are simple models, so to validate the results, we take advantage of our translational capacities to look for candidates coming from the models at patients, basically by genetic association, and more recently, we are starting to do something in the field of biomarkers. Infections are gaining a lot of interest in the field of dementia and Alzheimer's disease, and there is an increasing list of infectious agents able to trigger, including bacteria, fungi, viruses. There are some recent interest, for example, in buccal bacteria or others. Okay, a lot of them. And several viruses also. All of them can trigger degeneration by direct or indirect mechanism, mainly inflammation, which is very highly accepted as something derived from infection and having an effect in physiology. Among these infectious agents, herpes simplex virus 1 has the capacity to trigger both direct effects because it is latently present in the brain in many people, and so it can have both a direct effect on the cells, and besides, also it can trigger inflammatory processes. So it is is it associated with Alzheimer's? We have no the demonstration, of course, but evidences have been present since many years ago, and they are increasing. Some of them, and maybe the first one was this one by Itzhaki, who found that the presence of herpes simplex DNA in the brain, together with the possession of the genetic risk allele ApoE4, was strongly associated with the disease, whereas the virus alone or the allele alone was giving that risk. Okay, this is the first evidence. A lot of them has been accumulating. For example, some works have found peripheral markers of reactivation of herpes. These markers are found in blood. There are then markers, peripheral markers, genetic associations, and so on. From the experiments, it has been demonstrated that herpes is able to induce markers typical of Alzheimer's, like related with A-beta and with tau hyperphosphorylation, that the genomes localized in the brain of patients near the senile plaques. And recently there have been some studies on the capacity of A-beta, especially in oligomeric form, to protect cells and mice against the infection. This has been recently, supported by retrospective epidemiological studies nationwide, one in Korea, one in Sweden, more recent, that have found that these are cumulative incidence of dementia, not only Alzheimer's, but dementia in general. In the Swedish study, during the time, and it corresponds to people having been diagnosed with herpetic infection, no treatment, and these other ones are those having an anti-herpetic treatment. These evidences have led to the beginning of a couple of small clinical trials, proof of concept or phase 2, that are, as I'll tell you, ongoing. Okay, what are the data we have? And I'm sharing with you a brief historical overview of our functional models, neuroblastoma models of infection. We first studied the relation between the infection and typical ID marker, that is A-beta and tau phosphorylation, and we found that in our cell model, the virus was able to induce the accumulation of intracellular accumulation of A-beta on a time-dependent manner, and that this accumulation was accompanied by an inhibition of the secreted A-beta. That is a positive correlation between herpes and A-beta. We also found association with phosphorylation of tau. I'm not going to show you today. And what we found also was that this A-beta was accumulated was contained with autophagosomal vesicles. Yeah? So, in brief, we demonstrated that in the model, the virus is able to induce accumulation, specifically associated to autophagosomes, and this was accompanied by the inhibition of the autophagic flux. We also demonstrated that the oxidative stress was able to potentiate these markers of degeneration in the cells in spite of the fact that it inhibited the lytic cycle of the virus. Okay. To try to find what mechanisms were connecting the virus with the neurodegeneration marker, we performed a series of transcriptional studies focusing on the effect of oxidative stress on infected cells compared with that of non-infected cells. In this way, we got a list of genes characterizing what are the proteins, the genes that oxidative stress modulates in infected cells, but not in non-infected cells. This list was a big one, so we decided to cross it with the genes modulated in a cellular model, the same base lane, bearing the Swedish mutation causing monogenic disease, with the aim of finding some genes that were common to these two models. So this was a small list that, which functional annotation led us to the functions lysosome followed by lipid metabolism. So, and this is the main genes in the list, the major, the most modulated. So this suggested us that this pathway, lysosomal pathway, could be involved both in infection and mutation, so going to the disease to sporadic and familiar cases of the disease. So we then centered in this pathway, which is in where we are looking at. Okay, this is the lysosome system, and we are performing the studies, focusing on the study of AD markers, amyloidogenesis and tau phosphorylation, but also we are interested in the functioning and burden of the lysosomal vesicles in these cells, which are tightly linked to cholesterol trafficking in the cell. We have concentrated in 3 candidates that you have here. Among them, we are now working on this one, LAMP2, the lysosomal membrane protein, and a metalloprotease, which is a non-canonical secretase of APP, as we have recently demonstrated, and which is localized near the endosomal vesicles. So we are performing some functional experiment, as always, trying to connect these results with the patients. Okay. We have demonstrated that the virus is able to increase lysosomal burden accompanied by a reduction in the functionality of these organelles with a decrease in enzymatic activity. Besides, we have been able to show that the lysosomal, the endosome pathway trafficking is inhibiting by using a tracker of this pathway. So we have been able to demonstrate that not only the autophagic alteration we had seen before, but also the endosomal pathway could be altered in the models of infection, and this affects to the final steps of the pathway to the lysosomes, not only autophagosomes, but also to lysosome, also to the endosomal pathway. And that, interestingly, this was accompanied by an intracellular, we are not sure if in the lysosome or not, we are studying it, accumulation of cholesterol in the model. So, in brief, as panoramic of what are the characteristics we see in the models. Well, these are all of them. Alterations in AD-related markers, alteration in autophagy, also in endocytic pathways, and also alterations in how these cells manage cholesterol. All of this has been published, and not only this is again the list of defects we see in the models. All these defects have been suggested or demonstrated in the disease. There is a major alterations. There is a common point between the model and the disease. All of them pointing to the relevance of lysosome system. So in this scenario, we went to candidates in two ways. We are trying to demonstrate in the models if we can find targets of the pathway that hopefully could be actionable. And we are concentrated in these three, the two genes I told you before. They are highly modulated in the models, and they do more things that I'm going to tell you now. And also how we can manage or the infection or the neurodegeneration associated to the infection by modulating cholesterol levels or traffic. Okay. So we are looking for targets in the models, and of course, how relevant are these potential alterations or targets in the models? How are they related to the actual disease? And here we are starting, and hope to can do more on biomarkers related with the lysosomal system. As an example, one of these candidates, the LAMP2 candidate, which is a major lysosomal membrane protein, but that has a specific, well-demonstrated roles in the fusion of autophagosomes with lysosomes, but also has a major role in the export of cholesterol out of the lysosomes, but also have been demonstrated to interact with several viral proteins, not herpes protein, but influenza, even SARS-CoV-2. So we have started to dissect this relation between LAMP2 and herpes simplex by looking at several points in the infectious cycle in cells devoid in knockout cells, LAMP2 knockout neuroblastoma cells. We have found that the deficiency of LAMP2 had no effect in the early steps of infection. That is in the number of infected cells at early post-infection times, or in the expression of early proteins like ICP4, no difference between wild type and knockout cells. However, we see big differences at markers of late stages of the cycle, like the Here we have the expression of the late protein TC, but we also see a clear inhibition in viral replication, DNA replication, and also in the number of mature virions at the end of the cycle. So this is accompanied by a decrease in the neurodegeneration markers, A beta accumulation, tau hyperphosphorylation induced by the virus. We go to the next step, which is try to find if there is any direct interaction between a viral protein and the candidate that can help us to know how this is functioning, and also that could reveal any target to control the infection. We have found, actually, by mass spectrometry after following immunoprecipitation, some interesting candidates that we have no data by now. I will hope to have some of them for the next time I talk you. They include early proteins, but also proteins involved in DNA replication, and also, and it is interesting for us, glycoproteins that help the virus go to the nucleus through the endosome system. Okay, so in this picture of how is the cycle, the lytic cycle of herpes simplex infection, we have demonstrated that LAMP2 is able to is involved in the infections in its knockdown, reduced it. Probably this takes place at late stages of the cycle, and this inhibition is accompanied by a partial reversion of the markers induced by virus. Not only LAMP2, but also our second candidate, MMP14, is also able to inhibit the inhibition, in fact, very strong inhibition of the infection in the models. Probably take place at early stages using the same approach that for the other one, and also accompanied by a decrease in the markers. So all of these data, we think they are very interesting, but the majority of them comes from models that are far from a neuron, from a human neuron. So our current aim is we are starting to try to translate these studies to models more close to the neuron, or to the human neuron, because in the current model it is difficult, and we hope to then have the opportunity in these more physiological models not only to see the effect of lytic infection in the neurons, in differentiated neurons, but also to study the effect of the latency reactivation process, which is the more natural state of the virus in the human brain. Okay, now going to the, I think 5 minutes or so for the other part. Going to the studies we have been performing in humans to try to link the model with the disease. We have performed, and I have to tell here that we are very proud, very thankful to the organization, and more specifically to the HESCO consortium, because without these connections we will be unable. We have been unable, and we will be unable to perform this translational study. It is very, very important for us to be part of this organization. So thank you to the coordinators. I didn't want to forget it. So in this context, we have performed the study, genetic association studies, typical candidate directed to the candidates of our model. None of these ones give any answer, but in LAMP2, we found a SNP, a haplotype that gives a significant association, and this association was modulated by gender and by apoid genotype. So this, we have found a genetic association of the gene to the disease. Maybe not very strong, maybe controversial as all the genetic association, but we have found them in a Spanish sample. Okay, now I will tell you about the biomarkers design. It is our intention, and we are working on it, and we have a sample that is increasing, but this part of the work, preparation of the sample, as you all know, have been very difficult during the past almost 2 years, so it is going at his speed, not as we would like to, but it is going at. The design of this study is directed to early stages, mild cognitive impairment, even prodromic stages. Patients going to the clinic at the dementia unit of the La Paz hospital. These patients are being subjected to evaluation, both clinic and supported by biomarkers. We have a small sample now, not 100 yet. We hope to increase and to perform the follow-up studies that we have been unable to start by now, but we hope to. Okay, these patients will be studied, are being and will be studied. Okay. Sorry. No, the other way. Okay. Okay. In these patients, we are examining biomarkers at the periphery, both in plasma. We want to go to neural exosomes. We have not been able yet. And the other way, sorry again. Terminations in blood are also, and I'm showing you a small result in these cells. We are interested in blood mononuclear cells because they allow us to measure cholesterol and also what are the burden here, which are the burden and the activity of the lysosome system in cells that, although are peripheric, they are leaf cells. So I think we believe this material is interesting to study. Of course, we would like to, and we are collecting also cerebrospinal fluid samples to cross these studies of the periphery with markers at the cerebrospinal fluid. Okay, this is our intention. This is the design. We are going ahead, but in the meanwhile, we have been analyzing some archived samples available at the lab. So, in these archived samples, no, yes, oh, okay. These two ones coming from the Hospital La Paz, these are not the sample we are collecting now, but archived ones. And also thanks to a collaboration with the Hospital Peremata and the University Rovira and Virgili, we have relatively big samples for the leaf cells. Sorry. So what have we found? And these data are still unpublished, but I think they are worth to share with you. We have found that in the Hospital La Paz sample, this is as expected. There is more APOE4 carriers in the patients than in the controls in this way, but this other marker, the presence of detectable levels of IgM specific for herpes, is increased in the patients, but also not only in Alzheimer patients, but in MCI. We have been able to replicate this trend in the reduced sample, and even more interesting, when we analyze in the reduced sample what is the effect of the virus alone, APOE4 alone, or both together, similarly to the work of Itzhaki in the brain, we found this. We found that using IgM, specific for herpes, in the periphery, we get figures similar to that of Itzhaki. So this tells us that maybe these are a support that, based on these markers, that herpes could be involved, actually, and this is not new. There are evidences of other groups, also controversy. Our own data support the possibility that the infection is involved in the disease, that it can be involved in early stages, or relatively early stages, and that one can follow this in the periphery. Finally, we have concentrated in the measure of intracellular cholesterol in the leukocytes, peripheral blood mononuclear cells, based on a couple of previous evidences suggesting that these levels of cholesterol are altered in the periphery, skin fibroblast, or lymphocytes in this other work of Alzheimer's patients. So we started these evidences, together with the fact that we have found an accumulation of cholesterol in the model, in our cell model, lead us to start a study in which we have been using live cells coming from fecal purification that we analyze by using Philippine staining as the measure of cholesterol content, and also we analyze by immunoassays for the lysosomal proteins. I'm not telling you nothing about this part today. So this is the mouse is not obeying me very well. Okay. Further, in this analysis of Philippine, we took advantage of the multiplex capacity of flow cytometry to analyze the cholesterol content in these cells, but not only in the whole population of leukocytes, but in subpopulations representing the major lymphocytes and monocytes population in blood. What we have found, and this is the last slide, and this is preliminary results in the reduced sample, and is that there is a decrease in cholesterol content in MCI patients, also in Alzheimer's, compared to controls. There is another interesting result for me. I have not the explanation, but when we split the sample based on APOE4 carrying, no or yes, we found that this effect is major in the APOE4 non-carriers. This result is the same, basically the same trend, not only in the whole cells, but in all the subpopulations we have analyzed. The trend is the same, although we have found more consistent results in this population in which we have been able to replicate these results in the CD4 phagocytic macrophages subpopulation in the lab path sample. Okay, we have in the Reus sample. We see the same trend in all the subpopulation when we go to the lab path sample. This is very small, so this is very preliminary. We see the same trend almost only in the macrophages CD4 positive. So we continue these studies, and of course, it is possible that cholesterol in peripheral blood cells could serve as a marker. And what we need, of course, is increase the number, but especially to can perform longitudinal assays, follow the patients through the development of the disease. To the same, I repeat, and this is my last, I finish here. To the same, we are very glad to be near of this consortium, the other ones, because this facilitates to us very much the possibility to connect with people interested in these studies. So I finish, and that's all.